another question is lesson 9. All right.
Let's get started.
Yeah. Just gonna pull it up.
Sounds good. I'm gonna pull it up as well.
Transcription in E. coli is subdivided into 3 stages: initiation, elongation, and termination. So what DNA sequences are involved? DNA sequences are involved in initiation in E. coli. So I said the regulatory DNA sequences, promoter, terminator sequence. The promoter has a -30 of 5 site, -10 site, and a +1 site. And then what proteins are involved in initiation in E. coli, and that's sigma factor and RNA polymerase core enzyme, and they together make that up, the RNA polymerase hollow enzyme. For C, so what proteins are involved in elongation in E. coli? So it's that.
Huh? Right. Can we just step back for a little bit? So
sure.
on part A, it said which ones are. Yeah, 1A. It said what is involved in the initiation of E. coli. Why is the termination sequence involved in that?
Which one are you referring to?
Part A.
Yeah, but which one? What are you talking about, termination?
The terminator sequence. Why is that involved in the initiation of E. coli?
Let me look. Yeah, see, did you put that too? No?
Terminator sequence?
Yeah.
Yeah, I didn't think it was involved in the initiation step, so that's why I was kind of confused.
But why? Wait. Oh, yeah. Okay, so that's why you're confused. I see what you're saying. Yeah. I don't know, actually.
Like, I mean, personally, I think that's a typo because.
Yeah, they could be.
Why in the world would terminator sequence be involved in the initiation? I don't understand if she asked like, what are all the DNA sequences involved in, translation of E. coli? Then I understand that. But like in the initiation, I promise you, the terminator sequence is not involved.
Mm-hmm. Yeah.
So the rest of these are like the regulatory DNA sequences, which involve like, the operator and. Like other things binding to it to initiate transcription. And then, of course, the promoter where RNA polymerase, and then all the different parts. But I don't get the terminator sequence. I just don't get that.
Yeah, I think you're correct, though.
Okay, do you want me to ask her?
Sure.
Okay. I will make note of that, and I will ask her, okay? And I'll be sure to tag you into those emails, okay?
Sure.
Okay, awesome.
So see. So what proteins are involved in elongation in E. coli? So that's RNA polymerase core enzyme.
Yep.
What DNA sequences are involved in the termination? And there is the termination sequence.
There you go. That's where the termination sequence comes in.
So maybe that's just a typo. So I don't know.
It could be.
What proteins are involved in termination in E. coli? So the Rho protein and the nuclei, whatever.
Yeah, another question I had on that one was the Rho protein can sometimes be involved. Isn't that correct?
Yeah. Like, but otherwise, it just doesn't use the Rho protein, so it just forms the sense loops itself.
Right.
Right. So either the Rho protein is like making that happen, or it's not, and it's doing it itself. Where'd you go?
I'll be right back.
Oh.
All right. Sorry about that. Okay. All right, moving on to number 2. Okay. So it says E. coli produces a family of proteins called sigma factors. And it asks us what the function of the sigma factor is. So doesn't the sigma factor, like, is the part of the RNA polymerase that recognizes the promoter?
Say that again.
I said, isn't the sigma factor the part of the RNA polymerase core enzyme that that interacts with the promoter?
So they form together with RNA polymerase core enzyme. They form the RNA polymerase hollow enzyme.
Okay.
And it like binds to the -35 and -10 sites, and kind of guides the RNA polymerase core enzyme to the promoter.
Okay. So in that case, so when the sigma factor of the RNA polymerase core enzyme, when it interacts with so is the sigma factor on the promoter or is it on the?
So I believe that the sigma factor is attaching to the site first, and then it like the RNA polymerase core enzyme, it goes and attaches to the sigma factor.
Okay, so the RNA polymerase attaches to the sigma factor. Is the sigma factor on the promoter?
Yeah, the promoter is the -35 and 10 sites, right?
Okay, yes, it is. Maybe the sigma factor is separate from the RNA polymerase and the promoter.
What?
I'm just trying to figure out where the sigma factor is located.
So let's see.
Like, is it actually on the?
So -35 and -10, it serves as binding site for sigma factor.
The way that yeah, before times like.
Yeah, so that I believe so. That's how it is.
I actually think, based on the PowerPoint, that the sigma factor is completely separate from the RNA polymerase core enzyme and the promoter, but it's like kind of the middleman to connect the two together.
Yeah.
Okay.
But when the sigma factor and RNA polymerase core enzyme go together, they form RNA polymerase hollow enzyme.
Okay, so all together is called the RNA polymerase hollow enzyme.
Yeah.
Okay. Makes sense.
Cool.
So on part B, it says one of these sigma 70 is called the housekeeping sigma factor. What do you think housekeeping means? Okay, so for this one, I didn't really find it in the notes, so I just looked it up online. And housekeeping, the way I found it was that housekeeping means that this sigma factor is responsible for keeping the essential genes and proteins functioning properly.
Mm-hmm. Yeah.
So basically, it just helps keep all of the proteins on the genes and the promoters, right?
Yeah.
Okay. All right, cool. Part C reads, other sigma factors in E. coli, including the general starvation, sigma 38, nitrogen starvation, sigma 54, and excessive heat, sigma 32, sigma factor. Of what benefit is having multiple sigma factors to E. coli? And I just said that, these, so I said each of these sigma factors are responsible for the necessary ingredients needed to maintain the flow of catalysis in RNA polymerase. Each sigma factor has a specific job to do. So having additional ones can make it like more flexible, you know?
Yeah, I like that.
Yeah. The key reads the additional sigma factors allow an E. coli cell to quickly respond to stressful environments. Just for example,
Yeah.
E. coli encounters an environment that's low in nitrogen, so the sigma 54 will bind to the E. coli RNA polymerase core enzyme. So basically, depending on the environment they are, these different sigma factors can help like adjust for that.
Yep.
Which is, I guess, a little bit more specific than my answer. So, but that's fair. Okay.
Mhm.
Says sigma factor is a good example of a transcription factor. Provide 5 other examples: prokaryotic or eukaryotic transcription transcription factors. So I said like the alpha subunit. I said the beta subunit. I said the gamma subunit. I said TF2D. I said TF2B. I said TF2F, TF2E, TF2H, like all the
Well
TF2.
done. You got it.
Okay. Yeah, there's a lot of different types of transcription factors, you know. Okay. Like the
Good.
alpha subunit, beta subunit, and omega subunit, those are part of the RNA polymerase. So,
Mhm.
okay. You
So
want to
we
go?
learned, yeah. So we learned that E. coli terminates transcription using either the P-dependent or P-independent mechanism. So how are these two mechanisms different? So dependent, it's dependent on the raw protein and RNA or the like RUT interact. So they create a stem loop and then that's formed by GC pairs, which like makes hard like hydrogen bonds that are hard to break. And that slows the RNA polymerase, and pre-protein catches up with the RNA polymerase and releases the RNA polymerase and the RNA. And in independent, a loop is formed without it and does the same thing, it says. But there, the news say will add a U-rich sequence, um, and that forms weak H bonds with adenenes in the template with the template strands, so that they're like can
Yeah,
follow.
that's basically what I.
Yeah. So how are these two mechanisms similar? So they both create a stem loop, I said.
Yeah. I also mentioned that they both cause a delay in the RNA polymerase polymerizing the DNA sequence. Like
Yeah,
whether it was
that's
the protein or the hairpin loop that caused that. I also said that they both cause the RNA polymerase to disassociate from the DNA sequence.
mhm.
Pretty basic stuff, you know. Maybe I'm going a little too basic there, you know. It's like,
Maybe
oh, they're both use RNA polymerase. I don't know.
not. So for C, suppose you're studying P-dependent transcriptional termination in the lab. Suppose two ways you could interfere with the P-dependent transcriptional termination process. Think in terms of specific changes you can make to DNA or mRNA that would inhibit transcriptional termination. So since there is a GC-rich region, so if we mutate it, we could remove that region, like so that it doesn't have GC, so much GC, and then the stem loops would be removed. Or so if we mutate the root RUT so that the P protein never binds to it.
Yep.
And then for D, that was the same question, but P independent. There I said same, create a mutation in the GC rich region, just like on the other one. And the other one was a mutation in the region that NUSA makes, so that it can't terminate it.
Mm-hmm.
Did you say the same thing?
Oh yeah, basically, I did say around the same thing. Um,
Good.
I mean, my second way on the P on the row independent one was kind of a little shaky. I mean, it's both ways that are mentioned in the key are actually kind of similar. Basically, it says that you want to change the like either the GC base pairings or the AU base pairings.
Hmm.
It's like either change the GC rich so it doesn't form a dem loop, or change the A rich sequence so that way the DNA sequence doesn't have um,
Yeah. Do you want to do number 4?
yeah. Let's go ahead and go on to number four. But yeah, so okay, so for number four, when I first looked at it, I was very confused about the basal level. The Sister Mayo mentioned that she posted a video on how to solve number four. Have you had a chance to look over that video yet?
What?
I said for number four, you said that a lot of people were confused about it, and there wasn't really
No,
like,
no, no, not this one. It's
huh?
the number 4 on the applying genetics problems.
Oh, why
Yeah,
you jumped over on number four and the applying genetics problem?
that's the one she posted a video on.
Oh, it was applying genetics. Well, what about number four then? And
Here?
the teach one another, yeah.
Well, I mean, basal level, that's just the reading talks about that, like what's required for the basal level, and then basal transcription, and let's see, DNA, then, well, you first have your DNA sequences, and then the regulated, wasn't that like, yeah, like enhancers and silencers that can either upregulate or downregulate and stuff like that.
Let's talk about the basal level though. What does that mean?
Well, that's just I read it today, and the basal is basically the normal, I guess, default. I don't know.
Okay, so it's basically like the promoter and
Yeah, it's not. It's just transcribing in as it normally does. It doesn't have anything else that's acting upon it than those things. Like
okay, so,
the basic needs for it to transcribe, but if you add like
okay,
a enhancer, it's gonna go faster.
okay. I see what you mean. Fair enough. And this is B says what proteins are required for basic transcription in eukaryotes. So that would just be like, like the transcription factors and the RNA polymerase 2
For which one?
for 4B.
Yeah. Yep.
And C says
What did you
what
say
the
for?
for I said RNA polymerase 2 and then
No,
just
for A.
for A. What I say for A?
Yeah.
Just the core promoter, the Tata
Okay.
box.
Okay.
Okay. Part B, RNA polymerase 2, and then the general transcription factors like TF2D, B, F, E, H, all those.
Yep.
Okay. By the way, for these answers, did you just put the proteins, or did you put in the description like she has in the key?
No, I just put them in.
Okay, you just put them in. Yeah, that's what I did too. Well, she didn't ask us to describe them, so I'm just like, it says what DNA sequences are involved in regulated transcription in eukaryotes. So I said enhancers or silencers.
Yep.
Or
You
anything
still
else?
have a motor.
Still have the promoter. Okay.
Yeah, and promoter elements.
And then
Yeah,
the
and
promoter
enhancers and silencers.
elements, the key says.
Yeah.
Okay. All right, fair enough.
And then the D.
Yeah, D. Sorry. It reads what proteins are involved in regulate transcription in eukaryotes. Well, still the transcription factors and RNA polymerase too, but also I put activators and repressors,
Yeah.
and then the key
But
also.
what are they?
Go ahead.
But what are the activators and repressors?
What are the activators and repressors?
Are they like related to the enhancers?
They're actually more related to the operator. The operator. So activators and repressors are proteins that bind to. Actually, that's a little more complicated. Okay, so I've always been told that they bind to the operator, but here it says that
The operator.
the activator binds to the enhancer, and the repressor binds to the silencer.
Oh, so they are related?
Yeah, I guess they are, according to the key.
Okay.
I always thought, personally though, I always thought they bound to the operator, but I could be wrong, or in other words, it might be just more complicated than that.
Okay.
Maybe in other classes they told us like, oh yeah, it just binds to the operator, but in reality, it actually binds to the enhancer, and the repressor binds to the silencer. And of course the activator like enhances or raises or promotes transcription, whereas the repressor like reduces or inhibits transcription.
Yeah.
And then I'd also mention the mediator, which I don't know what the H to mediator is. What the heck is the mediator?
The mediator, let's see. Well, so it's also one of these general transcription factor. It seems like it's regulating interaction between RNA polymerase and other transcription factor.
Okay.
So I guess it does some sort of maybe a cascade effect. Like it does something to the TF2H that then regulates an RNA polymerase activity by phosphorylation. So I guess it starts some sort of cascade or something.
Yeah. In the key, it says that mediators are basically kind of again the middleman, like the sigma factor is, like how it's the middleman. Mediator is kind of the middleman between the promoter and the activator, or the promoter and the repressor. So, if the activator, if something binds to if the activator binds to the enhancer sequence, then it sends a signal which the mediator carries over to the RNA polymerase to either enhance it or reduce it.
Okay.
That's what it says. That makes sense, I mean, I've always wondered how, like, binding to the activator, like, enhances transcription or reduces it. I didn't really understand that. But if there's another protein called mediator that carries out that function, then that makes sense.
Okay.
Okay, you're up, man. 5.
So this question concerns the 3 major RNA modifications discussed in this lesson. So suppose a mRNA is produced by transcription. Which RNA modification occurs first? So the first is the 5' and 5' end capping. The second is 3' end polyadenylation, and then it's intron splicing.
Right? Is it always in that order?
Yeah, it seems like the reading said that that's happening in that order.
I know the 5'.
Huh?
I know the 5' capping and the 3' polya tail.
Yeah.
it comes before the intron splicing. That I knew, but I didn't know if there was. I thought they just both happened at the same time. Really, so
Yeah, the reading said it's sorry in that order.
okay. All right, on to part B.
Yeah. So ribozymes play an important role in RNA modification. What is a ribozyme? So a ribozyme that's like how self-splicing, like how they're cutting up themselves or something like that. It says are catalytic RNA molecules. And then which RNA modification process involve ribozymes, so that's intron splicing.
Yeah, they're basically RNA that acts as kind of like a enzyme to catalyze intron splicing.
Mm-hmm. Yeah.
Yeah, that's what they do.
So for C, what is the function of each of the following molecules in RNA modification? So SNRNP, it's a subunit of spliceosome complex, has ribozymes which splices introns. Guanosine nucleoside, it's a cofactor of group 1 intron splicing. CPSF, involved in 3' polyadenylation. It cleaves pre-mRNA 10 to 35 nucleotides downstream of the adenylation signal. And poly-A polymerase, it's making that 250 bases to form the poly-A tail. Guanylyl transferase, so it's involved in 5' end capping, cleaves GTP to make GMP and pyruvate. What's it called? PPI.
GCP is cut into GMP and then pyrophosphate.
Pyrophosphate, that's right.
Yeah.
So for D, did you have anything to say on C?
Nope, nothing. That's pretty much what I got.
So for D, it says that like introns are often identified by examining the results of hybridization experiment in the electron microscope. If a gene contains 2 introns, drawn an image that represents the result of a hybridization experiment when the template DNA strand is hybridized to the mature mRNA, followed by hybridization with the coding DNA strand. So I just drew like kind of what happens. Didn't really label much, but yeah, I would recommend to watch the video because it has to do with the one that you posted about, number 4.
Oh, okay.
Because this is what you're gonna learn there.
What they look like.
I've done the applying genetics problems already.
Okay, because I had no idea on this one.
Yeah.
I mean, I can show you what I drew, but like I don't think that's right.
Yeah, I drew something similar.
Oh, really? Okay, yeah. So okay, yeah. So we drew something similar. Like I had no idea.
So nothing too exciting there, but like just watch the video and you'll get it more.
Okay.
Cool. I'll stop recording now.